Welcome to Ask an Astrobiologist. I am Dr. Victoria Meadows. I'm a professor of astronomy at the University of Washington, and I also work as the director of the UW's Astrobiology program. And that is a graduate program where we actually train young scientists to be astrobiologists. And my other hat is as the principal investigator for the Virtual Planetary Laboratory at the NASA Astrobiology Institute. So, as you can tell from all of those roles, I spend a lot of my time doing administration of science, and so I am both a scientist and a science administrator. When I'm doing my science, I'm working on trying to understand how we would recognize and characterize habitable exoplanets. So planets found orbiting around other stars, trying to learn what their environments might be like, whether or not they're capable of harboring life, and doing all of that from, a very great distance, from the distance to sort of the nearby stars. So my research group, the Virtual Planetary Laboratory, focuses around a single question, which is if we were to find, say, a terrestrial type exoplanet, a rocky planet orbiting around another star, would we be able to recognize whether or not it was able to harbor life or it had life on it? And so the whole team works towards trying to understand what planetary environments are like and how life makes an impact on a planetary environment that might be visible over very large distances. So that's the scientific research we work on, sort of in a nutshell. On the educational side, as I said, we have the academic program at the University of Washington, where we take in graduate students for a number of different sciences and have them learn and work together, understanding sort of a broad range of the different sciences that feed into astrobiology and the interconnectedness of those sciences, and then learning how to work together on interdisciplinary projects. So astrobiology, as many of you know, is an extremely interdisciplinary science, and so a large part of our training is in how to work across discipline boundaries and make sort of, bigger discoveries because of that, because you're not working within your own field. So the other question I have here is, what does a typical day look like? And I was joking with Sanjoy that I didn't know what a typical day looked like because none of my days are ever typical. And I think that's true, and that's one of the things I really love about being a scientist in particular, is that honestly you don't know what's going to happen that day when you turn up in the morning, especially when you have administration in your science as well. And I think that's fantastic. I mean, I've been frustrated at work, and there have been many sort of different challenges at work, but I have never ever been bored. But if I can try and give you an idea of what sort of some days might look like, typically, I come in the morning and I check in with Tina, our administrator, to make sure there aren't any crises that have evolved overnight or anything that's come in that we need to address quickly. So that might involve something to do with the students, or something to do with sort of fiscal parts of the science project I run. And then the rest of the day might involve some teaching, if I'm teaching that quarter, meetings with my graduate students to go over their scientific research, and that is really the best and most exciting part of my day, because they often have great things to show and share, and so I really enjoy those. And then, of course, I have the team meetings of the research group at large, and so we sort of get together and also talk about what other research people in the team have been doing. And I spend a lot of my time nowadays, at this stage in my career, reviewing things, reviewing people's papers, reviewing people's proposals, or reviewing proposals that have to go out. So there's a lot of administrative and academic reviewing. And then at the end of all that, there's a small amount of time to work on science, typically not on any given day, and typically I have to kind of carve my science out in the evenings or the weekends, but I do enjoy sort of trying to keep up on that. And the summers in particular are great. But normally it's, it's kind of the sort of day. I think some people think scientists just disappear into a lab and that's all we work on. But, it looks more like a business and more like sort of, again, management and administration. So why I chose astrobiology as a career? Well, I kind of came to astrobiology sort of in a roundabout way, as you might imagine, because given my stage of career, astrobiology happened mid-career for me, in a sense. And so I was trained as a planetary astronomer, in fact, so someone who could observe planets in our solar system and try and understand what their environments were like. So it's not too much of a leap for me to sort of then try and apply what I've learned in that field to trying to understand what exoplanets might look like. So I started out, I did a PhD actually with my advisor David Allen in Sydney, Australia. And he was one of the very few scientists in Australia that actually did planetary science. And so I started with him on sort of more of an extragalactic type of project, but I managed to convince him that really he should let me do some planetary science. So in the end, I got to look at the lower atmosphere of Venus remotely using infrared spectroscopy and use that to peer down through the clouds of Venus and learn something about what the very lower atmosphere of Venus is like, including the abundance of water there, and that also has a measure on the greenhouse effect. So all of these were kind of laying the foundation for a lot of astrobiological themes pertaining to habitability that I eventually got involved with. So after I graduated, I went to JPL to work with David Crisp, and we worked on Venus and planetary science primarily. But while I was there, I talked to Dave about using his radiative transfer model and trying to get realistic clouds into it to try to understand the limits of the habitable zone, in particular the runaway greenhouse effect. So that was kind of a follow-on from the Venus work I did. And so, we wrote that proposal. It ended up kind of in limbo at the time because if any of you have dealt with NASA funding, you know that sometimes things go in there, but nothing ever comes out. So we weren't really sure what was happening with that. But while that was going on, I was asked to lead one of these NASA Astrobiology Institute proposals. I think this was in the second round. It was the second round of the call. And I was kind of put up to that just because I'd had these interests in potentially looking at habitability on planets. And so we put together that particular proposal, and wonder of wonders, it actually won. And so that's how I started my career as an astrobiologist, in the sense that, you know, I suddenly found myself being a NASA Astrobiology Institute PI and leading a group of people working on aspects of habitability on exoplanets. So it wasn't so much that I chose it as a career, I just kind of was positioned with sort of the right peripheral knowledge at the time when, you know, NASA was calling for more participation in that type of thing. And I think in a general sense too, I've always been interested in science fiction, and I watched Cosmos and Carl Sagan when I was younger and was very impressed by that. And that was part of what helped me decide to go into astronomy and to be interested in planets in particular. And so that's kind of the general, you know, way back background on that. Based on that introduction, you'll guess that I don't do a lot of my own hands-on actual research. So I tend to live my science through my graduate students and co-workers, which is still extremely rewarding to be able to do. But I'm actually, at the moment, I'm working on a very large review paper on habitability, which came out of the workshop we had on revisiting the habitable zone, actually a few years ago now. And I would like to get that wrapped up this summer. So I am working on actually an interdisciplinary synthesis of all of the aspects that lead to planetary habitability, and it will be a fairly sizable review paper that I hope to have submitted by the end of the summer. So I'm working on that. But other than that, my team is working away on various aspects to do with understanding the pale blue dot, in a sense. So, you know, Ty Robinson on my team has developed this fantastic sort of what we call Earth in a box model that allows us to visualize the Earth as seen from a distant observer. And so he's been working, initially he worked with the Epoxy team, who were a spaceflight mission that looked back at the Earth and took data, and so we worked to help analyze those data. But Ty and myself currently working with the LCROSS team, which, if you remember that was the spacecraft that did a few flybys around the Earth and then crashed itself into the moon. But in the process, it got some fantastic images of the Earth that we're also analyzing with our Earth model. And in fact, we are working together to help the LCROSS team calibrate their data, which is not something we would have expected from a theoretical model, but it's ended up being extremely productive. And so we're actually, you know, providing value added to the LCROSS data. So that's a bunch of things we're doing, and I also work on sort of the impact of flares on the atmospheres of extrasolar planets. That's with Antigona Segura and Lucian Wolkowicz, and that's incredible fun. You know, we put a huge flare into the upper atmosphere of a planet and try and figure out what that actually does to the photochemistry and the effect for life on the surface of the planet. So maybe I will finish there. There's a bunch of other things I could talk about in my group, and I will maybe go to the question. So one of the questions here is on the topic of cosmos. How do you feel about the new cosmos coming out with Neil deGrasse Tyson? Well, I mean, I think I'm kind of torn about that. I think the original Cosmos is a classic, and I think it's still, still relevant and very good today. On the other hand, graphics and special effects and everything have evolved so much that maybe it is in fact time for a new version of this that could potentially be, a little bit more visually arresting. So that might be cool. So we'll see how that goes. So the other question I have is, what are the advantages of being an astrobiologist versus a unidisciplinary scientist? Right. I think it's the interactions primarily, and also the chance to tackle questions that are so big that you couldn't possibly make any headway with them as a unidisciplinary scientist. So, within a given field, depending on when you come to that field, a lot of the really cool questions that may well have already been answered early on. You find that in fields, it's like people publish papers in the 60s and 70s, and you think, oh, wow, it must have been great to be the first people to figure out what the clouds of Venus were made of, or, that kind of aspect. But of course, in any field, as we say, research tends to open more questions than answers. There still are things to do, but I think what I find from astrobiology is I can tackle huge questions like, is there life on that planet? How do I figure that out? So it tends to push you really to the very forefront of sciences as they're coming out, and allows you to kind of be a pioneer and a groundbreaker, I think. And it's very powerful, and it's enormous fun because, I mean, you know what you know, but as you work through a problem with other scientists, you learn a lot. So it's a great way to sort of learn a lot and learn broadly about different aspects that affect the problem that you're working on. And often, something will just come out of left field, and that's the most fun of all, is, another scientist will say, oh, well, didn't you realize that, for example, methyl groups are always used in metabolism, or, something will come out where you think, oh, geez, that's really relevant to what we're working on, and I had no idea, and the person from the other field is like, well, that's kind of obvious. But it's not till you work together as an interdisciplinary scientist that you can put all these pieces together and get, a really powerful answer to a really big question. How do I balance being an administrator and a scientist? Are there aspects about doing both that you particularly like or dislike? I do like both, otherwise I wouldn't do it. But balancing, I'm not sure how good I am at balancing, but I am getting better. I'm learning to say no to a lot more things. Obviously, I said yes to this, but I am learning to say no, which is a big part of being able to balance things. But I mean, what I like about administration is being able to enable new research. I mean, there's a power in that is really kind of exciting and very rewarding in being able to get projects off the ground and to do these. And I happen to be, I think, reasonably good at administration. And so, that can be a very rewarding part of my job. It's also a way to get satisfaction fairly quickly on things. Some research projects can be very long and drawn out. But I do also really enjoy the research, and it's just kind of a nice mental break during the day to flip back from, administration into science with my students and back again. So I just find it a really nice and varied type of career. So are there any aspects about doing both you particularly like or dislike? About doing both together? Again, being able to flip backwards and forwards. If I was doing only administration, I think I would go absolutely crazy. So, I mean, being able to have the science and to be able to keep in touch with that thing that you're trying to enable, I think is really, really key. All right, so there are no departments of astrobiology universities. How does one find a job in the academic world as an astrobiologist? Okay, well, first I want to say that I'm really glad there are no departments of astrobiology. You may think that's a strange answer, but I was actually in a group looking at interdisciplinarity at our university, and people said, well, why don't we have a department of astrobiology? And why don't we sort of, to me, concretize, all of these different interdisciplinary things into their own departments? And I said, I didn't like that because I wanted the fluidity of being able to, in astrobiology, there are certain disciplines that we all now accept are part of it, but there are others that are sort of more on the fringe. Would they not be included in the Department of Astrobiology? So I'd really much prefer this sort of program structure where we can pull in departments as needed for these sorts of interdisciplinary aspects. So the moment I'm actually against kind of concretizing and trying to say, okay, put a loop around and say, this is astrobiology and this is what will be in this department. But that's an aside. But how does one find a job in the academic world as an astrobiologist? Well, there are many, many departments that are, I've seen this evolution, and now recognizing, in particular in my area, in exoplanets, there are a lot of astronomy departments that say, hey, we want to have an exoplanet scientist. This is really sort of an area that's expanding and growing, and we can see that young people, young would-be scientists are very interested in this area. And so I think you do have to, again, scour the job ads, see if they mention astrobiology as one of the things that they're particularly interested in. And I think you'll find that quite a lot of those are popping up. And of course, the other thing I would recommend, of course, is the NAI's careers page, where they often pull in specific jobs that specifically want an astrobiologist for that. The other thing, of course, is to be a stealth astrobiologist, is to come in with your main discipline, the discipline you were trained in. And then, once you're in an academic position, you can start doing, pretty much anything within reason. And so you can potentially expand out and bring your astrobiology to that department. And I know many of our students have gone out and done that sort of thing. Okay, hi Vicky. So from Rika, along those lines, have you seen that having an astrobiology background has helped early career scientists get jobs, or do you think there's a lingering prejudice against the field because it's so new and because of the damage done by the arsenic paper? Okay, I think, and I'll just get my head back on screen here. I think being an astrobiologist is definitely an asset. I've said this before, but when I was an NAIPI and we had postdocs who were astrobiology trained, a very large fraction of them ended up in academic positions, and I think it's for this reason, in that if you're trained as an astrobiologist in interdisciplinary science, you are very interested in lots of other different fields and lots of other different scientists and research, and that is very, very attractive in a faculty position. People hiring faculty don't want to hire a mushroom who's going to sort of sit in their office, not interact with others, and to work only on their particular area of work. When people hire a faculty member, they're looking for a colleague who's going to integrate into a community and be part of a departmental effort. And so I think a lot of our astrobiologists, are trained to be interested and inquisitive in what other people are doing. They're also excellent communicators in general. So by the time you're trained to talk across disciplines, you're very good at talking to and being understood, as well as asking the right questions when talking to someone from a different discipline. And so I've seen those aspects of the training really benefit, so whether or not the person gets tagged as an astrobiologist, they have that kind of a background. So as far as, prejudice from sort of some of the things like the arsenic paper, I have not seen that, but then I'm speaking from an exoplanet perspective, where I don't think that, that hasn't really impacted us quite as much overall. Might be different in the biological sciences, for example. But, I think definitely it's an asset, and I just see the students who are trained as astrobiologists go out and are just, I think, excellent faculty members. So how does one train to be an astrobiologist, particularly because few universities have astrobiology as a curriculum? Okay, so the first rule you have to learn in training as an astrobiologist is you have to have a grounding in a core discipline. That's absolutely crucial. And that's because that has to be the base and the foundation from which you have something to offer to other people as part of this interdisciplinary science aspect. So you have that, but in addition to that, you are then, again, trained or you push yourself to interact with others, to get involved in interdisciplinary proposals and interdisciplinary work. And I think, obviously, I will say that the best place to do this is at the University of Washington's astrobiology program, but then, I have to say that, but I absolutely totally believe that. But also at other, if, not everybody can come to the University of Washington, but if you're at another department or another institution, I think you can certainly still work on your core science, but again, look for opportunities to get involved in interdisciplinary projects, look for opportunities to take your work to other disciplines, volunteer to be, give a colloquium in another department on something you think might be tangentially useful to them, that sort of thing. So I think, and again, attend all the astrobiology conferences that you can. But that would sort of be sort of a do-it-yourself try and train as an astrobiologist, but it also certainly would help, of course, to go to one of the departments, one of the institutions that has this type of program. And there are several. There's Penn State, there's Arizona State, there's UW, and there are other departments across the country that are really starting to understand that astrobiology is good for attracting bright young scientists, especially those with sort of very high curiosity levels who like to be creative. And I think, there's still going to be a lot of opportunities. It'll come down to talking one-on-one with the person you want as your potential advisor and learning about the culture of that university. Okay. What are some of the biggest challenges associated with identifying signatures of life on exoplanet based on the biology we know? So, there are a lot. So one of the biggest challenges is going to be, and we say this even in sort of non-remote sensing sense, but if you're trying to determine whether life has had an impact on an environment, you need to understand what that environment was like in the first place. You need to understand what are the characteristics of that environment and how can I discriminate my life signal from it. So one of the biggest challenges is really going to be able to get enough of an understanding of the environment to know whether life has in fact tampered with that, has left its mark. And I think that's kind of an observational issue. If I've only got like one CO2 band and one O2 band, I'm not going to be able to tell very much about that environment. But the other aspect, of course, we have is trying to figure out, given the diversity of different metabolisms that we have, which of these are going to produce byproducts that will survive in an atmosphere for sufficiently long that we can actually see them remotely via a telescope, and which ones are going to produce the right spectral signatures. So there's still an awful lot of work to be done on looking at sort of alternative metabolisms and how those impact planetary environments. And at the moment, as you know, we're kind of limited to those metabolisms we know about. But another thing we're trying to do is looking at whether there's any universality to metabolic processes or anything that we think might be indicative of different types of metabolisms, just in a more generic sense. What kind of an impact, say, energy-wise, and I guess, Sandra, you're probably working on some of this. Are there any sort of taking one step up and just saying not just oxygenic photosynthesis or, chemoautotrophy or whatever, just taking one step up and saying, okay, are there sort of very characteristic signatures of metabolism as a general class that we can find? So these are some of the challenges that we're trying to work on. So Grasshopper has asked, what are the most intriguing questions in astrobiology we should encourage young enthusiasts and scholars to start pursuing? Oh, gosh. Well, there is a roadmap. I think so. So from my perspective, I think part of that is definitely biosignatures. I think we have been focusing on habitability. This is for exoplanets, of course, speaking from my perspective. We've been focusing on habitability, but I still think, again, there's an awful lot of work that needs to be done to push past this sort of, not quite dogma, but this sort of rut that we've fallen into of saying, we want to see oxygen because oxygen represents, oxygenic photosynthesis. I think that is maybe an extremely highly likely biosignature, but we need to start looking at other aspects as well and to figure out what sort of, again, are the probabilistic signs that we are going to see. What things are going to be more likely given a different planetary composition? So I sort of put forward that as one of my key questions. Okay, so what's the next one? As the subject of astrobiology expands, what other subjects are up and coming that could be a potential for study? Okay, let me think. So I think definitely, I think we still have a lot to do in the origin of life. That's still in a sort of state where, we really have, again, just a lot to do in that particular area. So I think the origin of life, the evolution of metabolism, the interaction of metabolism with its environment, and then how that leads to biosignatures, these are all, I think, sort of very interesting areas for astrobiology to be in at the moment. Okay, so Julia's asked, when do you think we will have solid technology to be able to observe spectral signatures interesting for life? Okay, so I think, our very first chance of that is going to be the James Webb Space Telescope. which may potentially be able to pick up oxygen signals, for example, for super Earths, or if new research we're working on is correct, maybe even potentially for smaller planets than that. And so JWST in sort of 2018, 2019, I don't know what its current launch date is, but sometime in the next, 5, 6, 7 years, we may sort of get a glimpse at that first capability. But that's going to be extremely challenging for JWST. It will probably take the entire 5-year mission time to be able to get sort of the required signal to noise on that sort of thing. So again, that's looking at 10 years. And that's our best bet. And maybe it's only one target. Other than that, there is work going on at the moment for a TPF probe mission. So that is that's a mission that's capped at about $1 billion. It's going to be either a coronagraph or a culture. We're doing the research on that right now. It really is more of a technology demonstration. And we're not even sure right now whether it can get down to super Earths. We hope it will be able to, and that's what we're trying to drive the mission requirements to do. And again, that will have the wavelength range to potentially pick up things like oxygen or methane for an early Earth type planet, for example. And that, I'm not really sure exactly what the timescale for that is, but I'm suspect it's sort of 10 years or more. And then TPF itself, the thing we've always wanted, we're probably looking at, another 20 to 30 years on that one. But that might sound a bit depressing, but on the other hand, it means that, there's still time for us as astrobiologists to get in there and get the science done to make sure that we understand what it is that we're going to be looking for so that we build the right instruments to look for the right signals, and of course to get up on the curve on how are we going to analyze this data, which we know is going to be really, really challenging when it finally comes down. So what atmospheric constituents can we detect with current technology on exoplanetary atmospheres? So with current technology, we're basically doing the super Earths and mini Neptunes. Mini Neptunes primarily. We don't yet really have a super Earth with an atmosphere that we can observe in transit transmission. And we are looking at hot Jupiters as well. So the sort of constituents we can detect are things like carbon dioxide, methane, water vapor, and those are kind of the big three at the moment that people are focusing on. And so that's basically where we are at the moment with those ones. Okay, so Julia, could you brief us on the current concept study going on debating between the next gen of exoplanet imagers? Well, it's basically, that's the probe concept study, and it's just started. So at the moment, the two things in contention are a TPF coronagraph and a TPF type occulter. But again, these are probe class missions. So we're looking at most sort of a 1.5 meter mirror and, a cap at $1 billion. So that's our challenge, is to try and get, the best mirror light gathering capability and the technology to suppress the light. So that's really where we are. So I semi-answered your last question. Perhaps you could describe the differences between the missions. Well, at the moment, it's just, basically how we're going to do the nulling. So whether or not it's going to be a coronagraph concept or whether it's going to be something more like the New Worlds Observer with the flying occulter in front of this spacecraft. So what technologies do you use to keep up to date with the latest research, as astrobiology is so vast? I use graduate students. They're fantastic. So that's being a bit facetious, but I guess I'm still old school. People send me papers. I look on ADS. My students bring to attention, papers that they've found. In group meeting, we try and share papers that we find are particularly interesting that have come up. You look on AstroPH, that sort of thing. So I guess I'm still using sort of old school type technology. I don't have any trawlers or anything out that find things for me. Am I optimistic that we'll find life in the next 50 years? I think, yeah, I am. As long as we can get something like TPF flying, and as I said, there's even a chance in the next 5 to 10 years with JWST that we'll be able to find something. So I think it boils down to, how common do I think life is in the universe? And this is one of those things where, we've got this one planet, we've got this one example. We don't know how common life is, but that's what we do as astrobiologists. We're trying to figure out, okay, how common is life? Is it everywhere? but not yet able to communicate with us. But I'm optimistic. It just seems, again, crazy to me that we would be the only example, given that we're kind of an average terrestrial planet in what's turning out to be a not so average planetary system. But still, I think given the possible habitats for life out there, it would be unusual if we were the only place where life evolved, or at least originated. What are your thoughts on the current science funding status and NASA drastically reducing EPO funding? Well, at the moment, just in general, of course, I think that we should be spending more money on science, especially in this country. That's just a huge, broad overview, broad brush. Is that really we have, I think, the capability to spend more money on science, but it just needs to become a much higher priority. At the moment, of course, we're dealing with sequestration and the budget cuts, to NASA as a result of that. So far, it hasn't impacted astrobiology too badly, so we're kind of quietly optimistic that we might be able to ride this out. We're under the same restrictions as all other NASA civil servants, but the astrobiology program has been working to try and make sure that those aren't of too much impact to the scientists who are being funded and the graduate students who are being supported under that. So, quietly optimistic at the moment we'll be able to ride this out. It's not as bad as the sort of previous years when we've had our funding cut by 50%, for example, and that was a bit of a blow. So this time was sort of 5% and just trying to deal with that. It's not quite so bad. NASA drastically reducing EPO funding and farming it out to the Smithsonian. I'm really not sure that's the right way to go, and I think there is some pushback on that, and definitely, what I would like to see is that the NASA EPO people understand sort of what NASA's about, the importance of our science and research, and have sort of the on-the-ground knowledge of what's going on. I would feel very uncomfortable about having an organization that isn't, part of what we do, being in charge of the education and public outreach. I would worry about sort of the connection between those who know and have lived and breathed and understand NASA science, and getting that message out to the general public. So Sarah, how do you think origin of life research and exoplanet biosignature research might best inform one another? I think, again, this might go back to sort of the universality of life's characteristics. If there's anything we can learn from origin of life about how life originates and comes together, that can then inform us on what sort of environments we should be looking for, for ones that would originate life, and also gives us maybe a handle on this idea of trying to find sort of a universal biosignature, something that many metabolisms produce that we could look for in particular. So I think, yeah. And the stuff that we can feed back as exoplanet scientists, we also look, VPL looks back to the early Earth and tries to understand the different types of environments that were there. If we can help you through any of our techniques constrain what those early environments were like, then we can feed back into, okay, this is what we think the very earliest environments were like. This is where life would have originated. So I think there's definitely a feedback between those two areas. So, Julia, when you talk about JWST, is that using the eclipsing method to find biosignatures, or anticipating an occulta will be funded eventually? JWST, what I was talking about there was transit transmission spectroscopy. So that is just using JWST alone with no extra spacecraft, and just looking at sort of light passing through the atmosphere of a planet as it transits in front of a star. The occulter, I'd love to see an occulter fly in front of JWST. I just don't know what's going to happen with that, and I know that we're not allowed to modify JWST in any way to support that type of a spacecraft, like not being able to have anything to help with the alignment or whatever, so it looks a bit challenging. But I mean, now we have this probe study where an occulter is in contention as a technology demonstration, so that is potentially one way where an occulter type mission could get a foot in the door. But again, that would not be on the same time scale as being able to put it in front of JWST. So I guess, Julia, at the moment, I really don't know what's happening with flying occulta with JWST, but the fact that we're sort of diverting all of our energies into this probe development does seem to suggest that maybe that's not on the table at the moment. So Julia also asked, could you describe some other exotic metabolisms that would produce a weird biosignature? Okay, so this was work done by Sean Goldman, who many of you know, and we looked at sulfur biosignatures and what they might do to a planetary atmosphere. And here's where we discovered something really interesting, in the sense that we were expecting dimethyl sulfate or dimethyl disulfate or methyl mercaptan or something would come out of those metabolisms and end up, being an exotic biosignature in the atmosphere. And what we discovered, and what we were kind of expecting, was that, these molecules are big, heavy, complex things, and they tend to get photolyzed, and their methyl groups get chopped off when that happens. And so we ended up with actually ethane in the planetary atmosphere from a sulfur biosphere, which was not what we were expecting. Now, DMS, DMDS, and methyl mercaptan were all there as well, but just not in quantities that were really easy to see in the spectra. But it turned out, because you could chop a methyl off a large fraction of these types of molecules, that ethane, which was produced by two methyl groups, turned out being, this biosignature. And we were not expecting that. So there are a whole bunch of different metabolites that come out. But again, to develop a biosignature, you have to have three things. Basically, it has to be produced by life, it has to survive in the atmosphere long enough to build up to detectable levels, and it has to produce a strong spectral signature in the area that your telescope is actually going to be sensitive in. So when you put all of those three things together, you can sometimes end up with things that you didn't expect, like ethane, for example, instead of methyl mercaptan. How much concern is there for false positives, particularly with the coarse resolution we currently have? Well, yes, false positives are going to be an issue. I mean, there's false positives, and then there's just not having enough information to be able to make a definitive decision about whether or not life is actually there. And I think, and I try to get this across to people, that when we're looking for biosignatures on other planets, really we're talking about a probability game here. We're talking about, okay, I've got these environmental characteristics, I have these odd things that I think might be due to life, but what is the probability that it's life and not some other type of process? And so we'll always be keeping that in mind. And false positives are definitely an area where we, again, prior to launching these missions, we want to know which of these signatures are truly unique. Does that never happen? Is it always possible that I'll be able to be fooled by something? I mean, oxygen could come from some other process, like, runaway greenhouse. So in all cases, we're trying to figure out not just whether or not this is a clear false positive, like, for example, the red edge reflectivity of plants. There are some minerals, cinnabar, other types of minerals or compounds that actually have, a typical type of red edge type reflectivity, and they're just, something you might find, an exotic thing you might find on the surface of a planet. So we need to know about those, but we also need to know about, okay, let's say I think I have a biosignature, but can I look for other things in the environment that might tell me that I'm being fooled, and that this is in fact formed by some other more exotic geological process or whatever. So anyway, you're very welcome, Julia. I hope we do get a mission that can detect life on nearby planets and get us direct imaging spectroscopy sometime in my lifetime, and hopefully definitely in your lifetime as well. Okay, cool. All right, well, thanks everybody. This was fun, and I wish you all the best, and I will sign off now and go and review something. All right. Talk to you later. Okay, bye.